Lapel.
Learning her principles of biochemistry. Chapter 1: The Foundations of Biochemistry. Genetic Foundations. Genetic continuity is vested in single DNA molecules. Hey there, welcome to our channel. Today, we're diving into the fascinating world of genetics and DNA. Ever wondered how genetic continuity is maintained through generations? Let's find out. First things first, let's talk about DNA. DNA, or deoxyribonucleic acid, is a long, thin, organic polymer that contains the genetic instructions used in the development and functioning of all known living organisms. Believe it or not, a single DNA molecule can be many centimeters long, containing a linear sequence of covalently linked nucleotide subunits, which encode the genetic message. To put things into perspective, a single page of a book contains about 5,000 characters, while the chromosome of E. coli contains about 5 million characters, nucleotide pairs. Now, imagine the textbook that would result from hand-copying this one a few trillion times. Genetic continuity is vested in single DNA molecules. The DNA that is the entire genetic material of an E. coli cell is a single molecule containing 4.64 million nucleotide pairs. That single molecule must be replicated perfectly in every detail if an E. coli cell is to give rise to identical progeny by cell division. There is no room for averaging in this process. The process of DNA replication is crucial for the transmission of genetic information from one generation to the next. It ensures that the genetic instructions are faithfully passed on, maintaining the continuity of traits and characteristics. The structure of DNA allows its replication and repair with near-perfect fidelity. DNA, or deoxyribonucleic acid, is a long, thin, organic polymer. It's the rare molecule that is constructed on the atomic scale in one dimension and the human scale in another. A molecule of DNA can be many centimeters long. DNA is made up of smaller units called nucleotides, which are the building blocks of the DNA molecule. These nucleotides are arranged in a precise linear sequence, and it's this sequence that encodes the genetic information. The DNA double helix is formed by two strands of nucleotides twisted around each other. Each nucleotide in one strand pairs specifically with a complementary nucleotide in the opposite strand. This complementary base pairing is crucial for the accurate replication and repair of DNA. Now, let's talk about DNA replication. Before a cell divides, the two DNA strands separate locally, and each serves as a template for the synthesis of a new, complementary strand. This process generates two identical double helical molecules, one for each daughter cell. But what happens when DNA gets damaged? Well, continuity of information is assured by the information present in the other strand, which can act as a template for repair of the damage. This remarkable ability of DNA to repair itself contributes to its near-perfect fidelity. Occasionally, mutations can occur in the DNA, leading to changes in the nucleotide sequence. While some mutations can be harmful, others better equip an organism to survive in its environment. This process of mutation and selection is the basis for evolutionary changes in organisms. The sequence of nucleotides in DNA encodes proteins with three-dimensional structures. The linear sequence in DNA codes for the production of a protein's unique three-dimensional shape, which is crucial to its function. The linear sequence in DNA encodes proteins with three-dimensional structures. DNA is like the blueprint for life, right? But did you know that the linear sequence of DNA actually encodes the three-dimensional structures of proteins? Let's break it down. DNA is made up of nucleotides, and these nucleotides form a unique three-dimensional structure. When DNA replicates, the two strands separate and two new strands are synthesized, each with a sequence complementary to the original strands. Now, here's where it gets interesting. The linear sequence of deoxyribonucleotides in the DNA actually codes for the production of a protein with a corresponding linear sequence of amino acids. This protein then folds into a particular three-dimensional shape, determined by its amino acid sequence. Once in its native conformation, a protein may associate non-covalently with other macromolecules to form functional complexes. But here's the kicker: the folding of many proteins is aided by molecular chaperones. So, to sum it up, the linear sequence in DNA encodes proteins with three-dimensional structures. It's like the ultimate recipe book for life. Thanks for joining us today on this journey through the world of genetics. Don't forget to like, share, and subscribe for more fascinating insights into the wonders of life. Until next time, keep exploring and learning.
Pharmapal.